A neuron spends its entire life doing exactly one thing that the rest of the brain can hear. It fires or it does not. So when you record a neuron for 2 seconds and it fires 10 times, what have you actually got? Like, not a picture, not a waveform, not really. You've got 10 moments in time, 10 numbers. That is a spike train. It is a list of times, and it is one of the most compressed descriptions of anything in biology. Think about what that compression buys you. A probe recording raw voltage generates something like a gigabyte a minute. You run spike sorting on it, which works out which blips belong to which cell, and what comes out the other side is a few hundred numbers per neuron. All that data boiled down to when did this cell fire. So today is about containers. Where do you put those numbers and what do you put beside them so that a dataset stays a dataset? Instead of becoming 40 loose variables you no longer trust. And at the end, we build a raster plot, which is a figure this whole field runs on. Lists. Start simple. A list is an ordered box of things. Square brackets. And commas in between. Here are the times in seconds that one neuron fired during a 2-second recording. Okay. 10 spikes. And notice they are in order, which matters because for a spike train, the order is the data. A list keeps order. That is its whole personality. Now let's look at this. You pull things out by position from a list in square brackets, right here. One thing to get used to. Python counts from 0. I've mentioned before in the previous videos. So position 0 is the first spike. Position 3 is the fourth. It feels wrong for a week and then it feels like nothing. The nice trick is -1, which means the last one. So basically, if you want to refer to the last item in the list, you can refer to it as -1. Like whatever the length is of the list doesn't matter. -1 always refers to the last item. You will use that constantly. A colon gives you a slice because that's one of the characteristics of lists. They are sliceable. A colon gives you a slice, a chunk of the list. Here is the rule to burn in. The start is included and the end is not. 0 to 3 gives you positions 0, 1, and 2. 3 of them and not 4. Everybody finds that annoying for a while. It does have a logic though. It means the numbers line up when you cut a list into pieces, no gaps and no overlaps. Here you can observe it. And lists can change after you make them. So that's another characteristic of the lists in Python. They are mutable. And lists can change after you make them, which is what made the append pattern from the last session to work. If you remember, we create an empty list and then we append values to that empty list as we moved forward in our loops, which we made them by 4. If you haven't checked the last video, please do before moving to this one. Now, a list is fine for spike times. It's genuinely bad for describing the neuron itself, because what else do you know about this cell? which brain region it came from, how deep the electrode was, whether the spike sorting was clean enough to trust it or not. You could shove all of this info of one cell of that into a list, and remember that position. That position 2 is a region. Please do not. You will forget by Thursday. A dictionary stores values under names, curly brackets, and each entry is a name, a column, then the value. Or basically, in other words, it's referred to as the key and the value. Exactly like an actual dictionary book. We have words, their meanings, their keys and their values. Look at what is in there. The ID, the region, the depth in micrometers, which tells you something real, because cortex is layered, and how deep your electrode sat tells you roughly which layer you were listening to, and the spike times sitting inside as a list. Containers go inside other containers. This is very interesting. This is very useful, specifically in this field. So, containers go inside other containers, which is the point of the whole session. You can add to a dictionary after the fact. Here I am adding a quality label, which in real work comes out of spike sorting and tells you whether this is one well-isolated cell or two cells the algorithm could not separate. Then keys shows you every field it has, which is what you run when somebody hands you a file and you have no idea what's in it. By the way, this structure is not something I made up for teaching. There is a standard format for neurophysiology data called neurodata without borders, NWB, built so labs can actually share recordings. Open one up and underneath it is this, labeled containers holding other labeled containers. Quick one, a tuple is a list that cannot be changed. Round brackets instead of square. Use it for things that should not move. An analysis window from 0 to 2 seconds, for example. A coordinate in the brain. 3 numbers that only mean something together. Why bother? Because if you accidentally write to it, Python stops you instead of quietly corrupting your analysis window halfway through a script. Second line there is called unpacking. You put 2 names on the left and Python hands out the value in order. Very common and very readable. From 0 to 2 seconds right now in this example. And a very useful concept. Nesting, which is where it actually becomes a dataset. Here is the payoff. A list of dictionaries is a dataset. Each dictionary is one neuron. The list holds all of them. 4 neurons here. 2 from V1, as you can see. 1 from hippocampal and 1 from motor cortex, M1, referred to as M1. Each with its own spike times. You can see the regions in front of them. For neurons, HPC, underscore, 007 is in hippocampus. And the second line, there is how you read a nested thing, okay? Dataset, open, right here. Dataset, square bracket, 2, gives you the third dictionary. Remember, from 0, 1, and 2. So 2 refers to the third entry. Then the square bracket ID in front of it reaches inside that dictionary for that ID field. You just walk in, one layer at a time, left to right. That is the logic. Now combine it with last session. Loop over the dataset and for each neuron count its spikes and computer rate. Beautiful. That is the summary table of a small recording in 4 lines. And notice it does not care how many neurons are in the list, 4 or 400, same code, same logic. That is the actual reason we do this, not like tidiness, scale and scalability. And my favorite part, right, the figure. A raster plot puts one row per neuron and one vertical tick for every spike. That's it. No averaging, no smoothing, no statistics. It is the most honest plot in the field because it's essentially the raw data drawn directly. Everything else you will ever do to the spiking data is a summary of this. I pull the spike times out of a comprehension, with the comprehension, pull the labels out with another, and hand them to event plot, which does the drawing. Right here. Beautiful. Okay, now look at the motor cortex. Neuron on the bottom row. Actually, the motor cortex at the top row. The spikes are not spread out evenly. They come in clumps. See? With silence between them. At the first glance, you might not see this, but if you take care of it, if you think about it, if you really look at it, you can see the pattern. They come in clumps with silence between them. That is the bursting. The thing we were classifying with a number last session, and here you can just see it. No threshold, no function, no decision. Your visual system found the pattern in about half a second. That's the importance of visualizing. To compare it to the hippocampal cell above, right here, like it below the motor cortex recording, which fires like a handful of times across the whole recording and looks almost empty. That is why you plot the raw data before you compute anything from it. A mean firing rate would have given you one number for each of those rows and thrown the entire data difference away. Okay, so for container. Lists, things in order. Dictionaries, things with names. Tuples, for things that must not change. And nesting them, which is how a real dataset is actually shaped. Exercise at the bottom of the notebook. Right here. Add a fifth neuron with spike times of your own and rerun the plot. Nothing else in the code needs to change. That is the whole argument for holding the data in the container rather than in loose variables. Next session, we hit a wall on purpose. So lists are great until you have 30,000 numbers and you want to average all of them. And Python start to crawl. Session 5 is NumPy. Where lists become arrays, everything gets about 50 times faster, and we build signal that carries us all the way through the session 9. This is also where this stops feeling like a programming tutorial and starts feeling like data analysis. So, notebooks and data are free on the GitHub. You can find the link to the GitHub repository in the comment. Link all available in the comments, so see you in the session 5.